So I'm going to start. So welcome everyone for the iSEE workshop. So we're going to describe today the package and the functionality. So this is the edition 2020. The package has been started in 2017, so we've got quite a few features to cover. And I'm glad to present that with Charlotte, Federico, and Aaron. All right, so the structure of the session, so we've got 55 minutes or so, a bit less now. So I'm going to start with a few introduction slides. We're going to set up the cloud workshop and then a bit of introduction to the interface. Federico is will pick up and present you the configuration. And Aaron will finish off with the future of designing new panel that you can do as well. So you've got the text chat and the polls in Pathable. You've got, you're welcome to join the iSEE Slack channel on the Bioconductor Slack. And the slides are available on GitHub. So we have an iSEE organization where you can find all the materials, including the vignette for this package. Okay, there we go. So yeah, just we have some friendly, scary, a couple of slides with a few bonus slides at the end. So please don't punch your screen if you're scared by the content. It's a funny feature of iSEE. I hope you'll enjoy them. So again, I want to acknowledge that this is a team effort. So Charlotte, Federico, and Aaron, probably familiar faces to anyone who's worked with Bioconductor for a bit. And yeah, so iSEE is designed for bioconductors. So that's what I'm trying to illustrate here. It really fits within the whole ecosystem of packages. In particular, it uses, it relies on the SummarizedExperiment, and the SummarizedExperiment is an extension of that, which we focused on mostly for the reduced dimension slot, which is regularly used in a lot of single cell workflows, but others as well. So I'm sure you're all familiar with the SummarizedExperiment by now, but I'm just going to go through. It's a really powerful container for data and very versatile. So you have the, if you see my mouse, at the center of the container, you have the assays, which are matrices, and you can stack as many as you want, one behind the other. The only restriction is that they have to be all the same dimension, so the same number of features and features which can be gene, and columns which are samples or cells. You have the metadata, which is stored in a slot called raw data. So that's where you can store all the metadata on your features. Similarly, you have the same metadata for the columns, so where you can store sample or cell-wise QC metrics, for instance. And then there's this extra slot for the reduced dimensions. So the rule there is that it has as many rows as the assay has columns, and each column in the reduced dimension is a dimension. So because this container is so popular, so widely used, I see through workflows, a lot of packages can interact with it from the data import all the way to the visualization. And so a typical single cell or really any workflow tends to go from the data import. So for single cell, you have droplet utils, or for more generally, you have TXT meta. Yeah, that's a wrapper around many different quantitation methods. Scatter uses the container to store quality metrics on the columns and the rows. Normalization, so SCRAN and ZIMWave, will add extra information in the various slots, including normalized data. Dimensionality reduction, so bias is singular. Scatter has some wrapper function, and ZIMWave populates the reduced dimension slot. Clustering is, so there's two popular packages and more, SC3 and Clustery. Again, the marker gene detection, you can store the fold change, the p-value of your marker genes as metadata on the features. And Slingshot is prepared for trajectory inference. And then downstream of that, you have, ggplot if you want to do visualization yourself. And iSEE sits at the end of this comprehensive workflow and just leverages the single cell experiment object to grab the data from the various slots. So the way it does it is because the container is so well structured, we know where to find the data. And what we do is through the interface allows you to access the data in each of the various slots to build different types of plots that I'm going to showcase in a moment. So the reduced dimension, the column metadata, the feature, and you can combine that into really powerful graphics. So yeah, that is what iSEE interface looks like by default. with a few tweaks, I'll admit. Yeah, and so I'm going to go through the interface with you to show you how the different what the different panels are there for, and how to use them. So the default panels, we have 6 plots and 2 tables, as you can see there. A little warning, a friendly warning, let's say. So we're not offending any other package out there. There's a lot of really nice and powerful interactive apps, which are listed actually on this repository. And before you go into creating your own application, I think I encourage you not just iSEE, but really to look at the solutions out there and see what they're doing, whether it's actually worth the effort. iSEE has been evolving for the last 2.5 years, and it's still growing, and there's a lot of effort that went into it. So it's really easy to try and reinvent the wheel and waste a lot of time. So we hope you enjoy the iSEE package. And yeah, again, I'll just mention that iSEE won the first Shiny contest, so for best technical submission. So this is how much effort we put into it. And we have this slide, so again, you'll find it on our GitHub repository with all the resources that come with iSEE. So the different packages, the manuscript, and a book to learn more about it. We can't fit it into vignettes anymore. Right, so taking you, I'll remove the full screen, and I'll take you to the Biosea workshop now. So right now, just you should have the link on the page, which is down here, the workshop Biosea. So you should be familiar with that now. And if I click the button, hopefully it shouldn't take too long. Oh, there we go, that was fast. And I'm gonna grab the password, although it should remember it. There we go. Okay, now we're gonna cheat a little bit to skip to the interesting bit. So in the chat on the page, Charlotte sent a little message to load the pre-processed data. So this is going to avoid us to go through a single-cell RNA-seq workflow and just grab the end result of the workflow. So I'll explain that in a second, but if you go to the chat and you copy this command, you'll get a populated single-cell experiment object that doesn't have just the counts, but also the cell and feature QC metrics. There we go, so it's also loading the package. Whoops, there. And if I ask the SC that I got, so it's got counts, normalized log counts, a lot of raw data, etc. So the workshop vignette itself will help you prepare this object yourself, but it does take 5 minutes, so we're going to skip over that. So yeah, you'll see it's just an example workflow. You load the data, you uniquify. So we transform the ensemble gene ID into gene names. We calculate some QC metrics for the cells and the genes. We exclude cells that have too much mitochondrial content. We calculate normalization factor and log norm, a log norm matrix. Modeling the gene variance, which allows you then to calculate reduced dimension, the PCA, TSNE, and UMAP. We use the SNN, so the nearest neighbor graph, to identify 14 clusters, and then using those clusters, we do differential expression between each of them to identify markers. So this is everything that's stored at the moment in the SC object. So you have the PCA, the TSNE, the UMAP, and everything I've just mentioned. Right, so getting to this, so that's usually what you would do in your analysis. And iSEE just sits at, oops, no, come back, there we go, at the bottom of this workflow. So, and at its simplest, we really try to make it easy for a newcomer. So you can load the iSEE package and create a fully functional Shiny app by just saying iSEE of your object. And if you store that in the app object, we have a Shiny app object.
Evan,
yes.
can you make your RStudio maybe a little bit larger text?
Yeah. Here we go. Yeah, and I don't need the right side. Very well done. Okay, so yeah, I mentioned here the reduced dimension, PCA TSNE, that's how I know I processed, and FDR, that's how I know I've got my markers in the object. Yeah, down here we have our Shiny app, and this is something that you just run by using this shiny run app of your app. So if you run it locally, you're going to have, it's going to be running on your local host, and you can also put that on a shiny server and deploy it and make it visible to your institute or to the whole wide world. Right, so the app automatically loads. So one of the nice things is if you don't say anything, iSEE will try to create one panel of each available type. So Federico will take a bit of time to show you how it's possible to configure the app so that if you don't care about certain panels, you can save time and space on your screen not having them. I'm just going to introduce you to all those panels. So yeah, we have the screenshot here. Another thing before I introduce you to the panel is it's also possible to launch an empty iSEE app. So the bonus point for that is like if someone doesn't have R installed or and you want to host the app, but without forcing them to use a certain data set, you can just put that empty shiny app and you will create two boxes where people can upload their own single-cell experiment object or summarized experiment object in an RDS file, and it will just import that file, launch the app. So you have a very versatile app on the web. What we're doing here is like we're launching an app that's fixed for a certain data set. Yeah, so at this point, you should have so from this section onwards, you should have an app with all the panels. And I'm going to go through the different ones. Do we have anything here? So it's important to know that it's not just single-cell experiment object. You can really put fax data, microarray data, anything that really you can fit into a summarized experiment. Because the class itself is very versatile, iSEE will just pick up the assays, the metadata, and everything. So you have all the detail here to read at your own leisure a bit later. So the first panel that I'm going to describe is the reduced dimension plots. So this panel is designed to essentially explore your single-cell experiment object. So if your object actually is single-cell experiment and contains a slot for reduced dimension plot, for reduced dimensions, iSEE will detect that and create a reduced dimension plot. If your object is a summarized experiment or DSC data set that doesn't have that slot, it will just not include the panel. So here in the screenshots, you can see that we already pre-configured the panel to be colored by cluster, and it's a t-SNE instead of a PCA. So what iSEE allows you to do is open, you have different collapsible boxes here, one to control the data aspects, another one for the visual aspect, and a last one for selections. So the PCA, the type of reduced dimension that you want to display, we consider data. And so if you remember, we've computed a PCA, a t-SNE, and a UMAP, and that's exactly what iSEE detects in there. So if we want to select the t-SNE, all you have to do is click, and it will just update the plot. You can also, if you have more than 2 dimensions, explore the further dimension. And so the example on the right is also colored by cluster. So this is a visual aspect that doesn't affect the layout of the points. And the cluster is a column, a sample metadata. So if I select color by column data, and then in the drop-down menu, it will detect essentially every column in the column metadata. So, and it comes with auto-completion. So if I start typing cluster, it just really quickly narrows it down. And when I select it, the plot updates, and I have literally the same plot as the example there. So this is how you interact with this panel and pretty much every other panel. So what really changes between different panels is the data aspects usually. So the column data plot is a type of plot that focuses on column data, as the name states. So that means that the y-axis will immediately start looking into your column metadata, and you have the same selectable box, selectize input, where you can control that. So here, the example is the log10 total count for each cell. So essentially the library size in log10 space. And if I start typing LO, I can narrow it down. So now that's one violin because the x-axis is not linked to any aesthetic variable. So what we have on the x-axis in the example is the cluster. And that is, again, x-axis, it's a column data. And if I type this CLU, I have my cluster. So it's a bit more compressed because of the panel width. If I stretch it a little bit, it would start looking like the example on the right. So yeah, that takes a bit of, there we go. Okay, I'm just going to narrow it down again. This is for the column metadata. So similarly, we have the same for the row metadata. So here it's the number of cells in which each feature is detected versus the log10 total count for that feature. So I'm going to go a bit more quickly and say, so this box looks at the raw metadata. So you will see, because it's a different panel, I don't have the same menu, the same values. So I'm going to go for the detected cells and the log10 total, but this is now by feature. And I get exactly the same plot as there. So I'm going to start collapsing the boxes a bit. A really powerful visualization is heat maps. So the heat map is a bit of a painful feature to implement. It's very difficult to make it interactive, but it's very powerful in meaning that you can visualize multiple features in multiple cells. And we have two different ways to control the features shown in a heat map. One is to send a selection from another panel, and the other one is to essentially edit manually the list of features that you're interested in. So this is with this edit feature names button, and here we don't really care about this microRNA. So if I take the example in the vignette, I can start typing my feature. CD68, CD19, and CD4. And I'm going to also put a wrong gene name because we have this little button here that says validate gene name, and it's going to comment out everything that's not a proper gene name in your, a feature name in your data set. So you can order alphabetically, or you can clear the editor if you don't want to do things manually. But once you have a list that you're happy with, you can just apply, and you're going to get your features in the heat map. So it still doesn't look quite what we have on the right, because we have the log counts centered. So this is a visual parameter because we're doing it on the fly. And the transformation here, we have this checkbox, and you can center. Once you've centered, you can scale, but in this case, we don't really care about scaling. It still doesn't look quite what we want, because the example here we have is put annotations at the top. So the column annotation, we've grouped our cell by clusters, so which is down here. And once I start typing that, then we start having this blob of yellow and yellow here and there. So yeah, you can do that. You have a lot of different features that would take hours to go through the entirety. I'm going to speed up a bit so I can hand over to the others briefly. So the same way, the next plot is the feature assay plot that essentially focuses the y-axis on a feature. So if I browse the interface, it's up here. So here it's CD27. So if you go in the data parameter, you can say that the feature to map to the x-axis is CD27. There we go, we have it there. And it's on the x-axis, again, we just use the cluster assignment. And one more thing is that with the visual parameters, we also color by cluster, which is a column metadata. And here we go. So we have the same with the two blue clusters here that have very little expression of this. And the same way you have a feature assay plot, we also have a sample assay. So in this case, your y-axis is a cell, and each dot is a feature for that cell. So what you do is here it's cell 2 on the y-axis and cell 1 on the x-axis. And that's how we got this plot. So we've got a nice correlation between those two cells, probably the same cell type. There we go. And the last two panels that don't really need any introduction, the raw data table is literally a visualization of your raw metadata, so which is up here. So you can scroll to the right, to the left. You've got filtering boxes at the top, and you have the column data table. So those tables are usually very nice if you want to select a gene and color, for example, this by so you can take the reduced dimension plot and say, I want to color by feature name, and this feature name has to come from the raw data table 1. And here we go. So I've selected.
Wrong one, Kevin. You picked the raw data plot.
Haha. Oh, row data table. There we go. So, and once you select it, actually, iSEE is trying to help you in telling you, yes, I am receiving information from that plot, and the other plot knows that it's selecting, sending information. So now I've got FUM here, I've got ORF there, and you can go through all your favorite genes. I think it's time for me to hand over to the others.
Then there's one question that's related to this. So some of the questions we will come to. So downloading plots we will come to ICU. But what if a column in a column data is a list column? So then what will happen?
We do, so Arnold, correct me if I'm wrong, but I think we were flattening the tables. I'm not sure if that's still the case.
If it's not an atomic vector, it just doesn't show up as an option in the in anything really. So yeah, I wouldn't really know how to handle that. You mean when you mean a list column? I'm interpreting a list column as in a column where you have one list per row of the data frame. That's how I understand it. So in these default panels, at least, they don't know how to deal with that, so they'll just ignore it. Of course, you can write your own panel that knows how to deal with it, but
yeah. Nested data frame are handled in a way that they just get flattened out, so all the components of the list or of that data frame, for example, would ideally.
That used to be the previous behavior, but in the latest version, that's no longer the case, I believe. Just to keep things simple, because yeah.
We had it at some point, but I felt like I've seen it disappear in recent updates. Yeah. So speaking of recent updates, I just mentioned, as long as I've got the interface, that yes, it is possible to download the panel outputs. So this is the menu at the top here, this little icon for downloading, and this button, download panel outputs, and you will detect all the panels that you have up there, and you can select if there's some of them that you don't want to download. And once you're ready, you just hit the download button. It's gonna generate them as PDFs and CSVs and just zip it and upload it. So if it's running on a server, you will download the file automatically. Well, and the same thing if it's running locally, really. Yeah, so that is that. You can, one of the most powerful and friendly feature that we had from very early on is you can download the code that generates all those plots. So one, it's nice to have the plots, but it's better to have the code that generated those plots so that you can play, it's first reproducible, and you can also tweak it and put your own colors on top of it. We won't hold it against you. So if I grab some code from here and I ran it, I would literally generate every plot. So that is that. And I think, yeah, we have, you can run a little tour, open the vignette, and get more information about iSEE and us. Yeah, maybe, I don't know. Federico, are you ready to take over?
I am. Are there any other questions, Charlotte, before we move on?
I think we will come to many of them a little bit later during the workshop. So I think you can continue, and whatever we don't get to during your well, what you're going to talk about, then we will answer them afterwards.
Okay, I'm going to take over then now. And Kevin left us with this SCE object that I'm just say typing for completeness. And whatever we did is just this iSEE of this SCE object, and that gets started with our app that I'm going to just open up here and make a little larger so that it fits on the screen. I'm going to probably just hide the thumbnail so that you're not in there. So what we can do with that is that Kevin mentioned that we can configure the panel of the app in the way that we want. And that we can do that pretty simply with this organize panels button. And what you can do is that you can take these movable elements and then just probably rearrange them in the way that we want. Of course, now I just don't want the heat map, or I don't need the column data, and I don't like to have sample assay plots messing around. And I would like to have these reduced dimension plot a little larger. I would just click on apply settings and boom, iSEE just does it for you. So the other panels are just below. I'm just gonna revert back to the initial configuration because it's pretty practice to have these 3 panels in the first row. And yeah, what Kevin also showed, you can, some features were already demonstrated here, but you can have more. You can have more reduced dimension plot. You can have how many as you want. And if you want to restart the app in such a way that you can get this exact configuration, what you just need to do is that to display the panel settings and then copy paste that, and then it just becomes your initial object that you will have to pass in your call to iSEE SCE initial equal to initial. So what you can do is that whenever you are done with rearranging or arranging your panels, you can move on and then start enjoying the whole graphical visualization, so the whole displaying part. It's already been shown by Kevin. So in the data parameters, you can, for example, you can switch from the PCA to the t-SNE plot, and you can do so just with a click. And you can have, as I said, you can have more. So we can put two of them close to each other, so we can compare, for example, PCA and t-SNE. And otherwise, I'm just gonna probably touch point-wise on each feature. Otherwise, it's gonna take too long. So we can display, for example, the expression level of any gene of our interest. So let's take CD79, B in this case, which was a marker for B cells, if I remember correctly, or CD79A, was it? Yes, so sorry for hiding you the autocompletion. And that's marked in this one. You can also display by the column data, as Kevin already shown. So the cluster just can show up, and that's clearly there. Or we can do more. We can, for example, have, let me just go there. You can change, for example, the shape, the size, and the point property. So if you want to just say shape by the cluster and combine that with the information from other metadata. So we keep the shape information and we just color by the feature name that we had. It's not so informative now, but you can say, imagine all the possible combination of yours and then say, play around. You can also change the opacity of the points, which can be interesting if, for example, if you want to have some kind of indication of how the density of points is distributed. And maybe I just make it a little smaller. So probably you can read, right? If it's like that. I'm just gonna have it so in such a way that you see that it just gets updated live. So the points become faint and fainter. So then we have a column data plot, for example, that Kevin already demonstrated. So I would have probably skipped to the, say, one of the main characters in our toy, which is the feature assay plot. The feature assay plot is intelligent enough to understand, for example, a couple of things. How the data is provided. So if you provide, for example, a categorical variable versus a continuous one, it will display violin plots, which are nice to see the distribution of that feature. If you display, for example, let me just get there. If it's a continuous versus a continuous, you will have a scatter plot. And if it's a categorical versus a categorical, you will have a so-called Hinton plot, which is nothing but the representation of a table in a visual format. So you can, for example, pick the information of a gene of yours. So this CD3D, this is the general distribution of across all the cells. You can split that up by the cluster. Or you can have that information, the CD3D, you can plot it against another gene. For example, CD40LG. And what you can do is that you can, for example, take these dots, brush with your mouse, so click and hold, and then this selection can be passed to other panels, all other panels basically. So this can be controlled by the selection parameters, and you can receive the selection from the feature assay plot 2. And this highlights the dots in my plot that are passed from the other panel. You can imagine how this can go on and on and on. When it becomes complex, there is still a nice indication of how your panels are linked to each other. So this feature assay plot 2 is passing information to this feature assay plot 1. It's not so mega visible, but apologies for that. But it also displays you all the panels that you have. So the linking and transmission is there. What is also nice is that we have a clunky yet oops, okay. That was the crash that we would not expect. So I don't know what I just did to crash, so we will follow up later on that. I just restored the app.
I think you just need to click slowly. You need to wait after the click that the point appears.
Okay.
Because it has to contact the server to draw the point.
I'm gonna try this time. So as long as you click slowly enough and wrap up, so you see that we just now selected a number of points with a non-rectangular selection, which is pretty handy if you ask me, because otherwise it wouldn't be possible to capture this strange shape, which is not showing now. And now I'm patient, but let's see if it works now. Okay. Oh.
Yeah, RStudio promised us at some point to look into it.
Okay. Yeah. Now it's even recorded, so we will have some leverage for that. Okay. Kevin already give you a sneak peek in one of the features I like most. So the fact that whatever you've been generating live, you can just click on that code, on that extract the R code button, and then you just Control-A, Control-C, and Control-V, just put it in your report or script, and then you generate one-to-one all the plots that you have done. I'll skip from here. So I just followed some parts of the vignette, but I skipped some of the parts because otherwise it really does take too long. And we just want to give you a nice appetizer of what you can do. There is a lot of what you can do with filtering of tables. You can save and restore the state of the app. So the state of the app is what you can, for example, know from here. The panel settings is what you had. Custom panels are more or less mostly Aaron's territory, so I will not enter into that. But what I really like to show you is, for example, a quick tour of the app. When a powerful application, now it's, of course, it's only enabled for the Allen dataset. So, oops.
We do have a little chunk of code with a demo tour. Where is it? If you search the vignettes, there will be a tour.
Yeah. Let me just get to there. So sharing and communication. So here.
There we go. Yeah.
So I'm just gonna close this one. Ooh. It's not liking it. I don't know why. Oops, sorry. So when the initial is not there.
Oh, that depends on the panel.
Let me just do that.
It's all right.
And then up, not apply, app. So when an application becomes complex and powerful, it becomes also harder for you to get user to do things. And we believe strongly into learning by doing as a nice application, as a nice pedagogic model to do so. The tour just does nothing but highlighting some elements of the app that you can just say click after each other. So either by clicking on the next button or on the arrows, and you can basically guide the user throughout the different elements. It's also that you can also link them nicely if you try to probably tailor a story around what you are showing. And we think of this as a possible feature when you want to deploy your dataset with iSEE, and then basically try to reproduce, for example, the panels of a figure of your manuscript. Something that people have been doing, so, and they didn't contact us, so it means that it's probably even feasible. Okay. People that know me know that I can talk for long and long and long, but I'm not gonna do this now, and I'm gonna pass it over to Aaron for giving us a nice introduction on the custom panels and on more.
We take maybe a couple of questions. So there are a couple of questions related to the type of data you can put in. So it's possible to use iSEE on transcripts instead of genes. And there was something else. But yeah, so you mentioned iSEE can handle any kind of features as long as data fits summarized experiments. So that would apply to different annotations than ensemble as well. So basically, yeah.
Pretty much.
Yeah. So there
any
is
type of data.
At some point, I took some variants, data, I ran a PC on it. I mean, it's not the most exciting when you got binary data. It's the plots are a bit more, the variant plots and everything get better with continuous data, gene expression. But yeah, anything you can think of and fit in there. We have a range of example on that came with the manuscript that we also distributed as Docker containers. And I think they're still hosted on the Marini group web server, Shiny server.
Whatever fits into your rectangular, expanded, annotated, summarized experiment object can be done.
Yeah.
We made some examples with the diamonds data as well. Right? It doesn't have to be biological data.
I'm gonna, so while Aaron is doing that, I'm gonna fetch my stupid demo on the diamonds dataset.
Right. Can people see this? Okay. Yes. Okay, so I'll talk about the custom panels. So there's two layers of custom panels, and let me just scroll all the way down to the bottom. Keep on scrolling. There's two levels of custom panels. There's a sort of quick and dirty custom panels, and then there's the sort of more properly engineered custom panels. And I'll just go through the first quick and dirty. Whoops, the quick and dirty ones. So sometimes the default panels don't quite serve what they don't do what you want to do, but you still want to use iSEE and provide the rest of the boilerplate for setting up the Shiny app, right? So you still want to use iSEE for the parsing, for the managing the selections and having reproducible code and all that. So there's a way to have your cake and eat it too in terms of defining your own custom functions. And then putting them into iSEE to generate the visualization. So one example here, where am I? One example here is, for example, in this case, maybe I'll show this. This is a bit nicer. One example, yes, the formatting is nicer. Yes, where was I? Yeah. So one example might be that you want to write an app, you want to write a panel. You want a panel that receives a selection of points from another panel in the app, and then performs a dimensionality reduction on that subset of points, right? So it's like the, if you're doing single cell data analyses or really any data analysis, any exploratory data analysis, it's just like taking a cluster and then applying more clustering on it to identify subclusters. So we can do that in this app, not by using the default panels, because the default panels don't know anything about, clustering or whatever, right? But we can do that by just writing a new function that generates a plot of a reduced, of a dimensionality reduction result of a subset of your data set. So in this case, I've created a function here. So all this function does is it takes a summarized experiment object. It takes a row selection, which is optional. It takes a column selection, and then it's got a few other parameters that control the how the dimensionality reduction is done. Just ignore this bit for a second. It just makes sure that the function behaves properly if you don't select any columns. But other than that, you have it diverts to 3 different functions depending on which dimensionality reduction method you pick, and then it just applies that function and then creates the plot. And that's really, that's all it does, right? It's a function that takes a summarized experiment and some row and column selections, and it makes a GG plot at the end. So let's try running that. Let's try. Oh, yeah, okay, this defines the function. Okay, and before I actually show you, let's say that. Okay, no, I do show you here. Okay, good. I wish this would go out of the way. Okay, and then let's just run that, and then let's get this. Are you still seeing this, by the way? Is this? We see
the RStudio session. When you launch the app, I don't see it now, but
okay.
We see the RStudio.
Okay. I don't know how to change.
Just kill the session. If you run app with launch.browser, true, it will open it in your web browser instead of trying to launch a pop-up. I suspect.
All right, I didn't understand any of that.
Oh.
Right, is this better?
It's reloading. Oh yeah, there it is. Yep, we've got everything.
All right, right. So yeah, hopefully you guys can all see this. You know, this is the same reduced dimension plot as we had before. So this is what Kevin talked about. We also got a row data plot here. We've also got a custom plot, right? And right now it's just saying, you know, well, I can't really see what it's saying because, you know, Kevin's face is obscuring it. It says no column data selected. Yeah, okay, good. So all right, so let's try selecting something. I'm gonna convert this into a tsne, which I think is classier than a UMAP, but whatever.
No, you didn't feel that.
Not in flames.
Sorry, what?
Flames.
Anyway, so let's say I select something, you know, okay, and then it decides yes, okay, good. If I select something, then and eventually, what you'll see that okay, in this case it's pretty boring. Let me do a more fun selection. Let's select this. All right, let's see what happens here. So yeah, when I select something, what happens is that the selection then gets transmitted from this plot to this panel, and then this panel does a new dimensionality reduction on, you know, the points that I selected. So you can see that, hey, you know, I've now got, you know, two things here, which correspond, I assume, to, you know, these two populations that I've selected. And the cool thing is that actually the system will automatically detect the other parameters that you pass into the function. So if you remember, where was it? If you remember, our function had this ntop and scale parameter, and actually what happens is that you know, when the panel sees those functions, it'll actually generate UI elements corresponding to those, you know, to those things, right? So I could just, like, you know, unscale it, and then it'll recompute the you know, the tsne without that scaling, right? At some point. Yeah, okay, there we go. So this is pretty cool. And, you know, if you were so inclined, you could also say, hey, you know, what parameters do I have here? Do I have variance? Okay, well, I guess not. You could say, hey, you know, I actually want to, you know, control the genes that I use for this. So I'm going to say, hey, you know, I'm going to select, you know, just these genes that probably aren't really all that interesting, but, you know, whatever. Let's just select these genes to do the thing. And then the dimensionality reduction will be just done on these cells and these genes, right? And this is all controlled by the fact that it receives a row selection from the row data plot, which is this guy here, and it receives a column selection from the reduced dimensionality, the dimensionality reduction result, which is this guy here. So that's pretty cool. That's a easy way to get these custom, you know, so, you know, you can put whatever you want in here, right? You can put, as long as it generates a ggplot, you know, iSEE doesn't care. So you can put whatever you want. What else did I have to say here? Nothing, I guess. Okay, good. All right. So another example. So that's for a custom plot. But what if, you know, you're not doing a custom plot, and you want to do a custom table, right? It's like, so, you know, we can do that too. So, you know, ignore all that. So, you know, in this case, we're getting now I'll switch back to this. In this case, we're setting up a custom differential expression, a custom function that computes differential expression statistics between the selection of points that you have versus, you know, the things that you didn't select, basically. So you can do anything here. You can do edgeR, you can do limma, DE-seq, you can, you know, do your t-tests or whatever. Right now, this function actually does something really simple. It just computes log-fold changes. But, you know, you can do anything arbitrarily complex that you want to do inside these functions. And then if we run this, oh yeah, whoops. Okay, let me stop that. If we run this, so what's this other function that I had? Oh, and I've also got a thing to make a custom heat map, and we'll have a look at that in a second. But if I run this and set this up, and then I run this, and then I open it in browser, and then I move this here, and then I wait. There we go. Yeah, all right, Fed, I'll make a UMAP for you. Fine. Yeah, so we've got all this, and even if you don't have any clusters, you could say, okay, you know what? I want to know. I want to know what these guys are, right? I want to know. I want to know what's up in these guys compared to the rest. And oh, do I have to do a row selection?
No, I think you've got to do a reference selection.
Oh yes, yes. Sorry, I got confused. I got confused with the other panel that does the same thing. Okay, yes. Whoops. Right. So yes. So let's say I save this. So what you can do, another cool thing that you can do is you can save selections. So you can just see it. You can just see it, right? It's this one here. So you can actually save a selection, and then you can make other selections. So you can make these disjoint selections that don't necessarily, aren't in the same space. But anyway, so when I say in this case, in this particular version of the panel that we've sort of hacked together here, when you save a selection, it gets transmitted to this custom panel, custom table, and then you can see that all genes have a log fold change between all cells in the selection and all cells that are not in the selection. So we could okay, I guess it doesn't like that.
No, you just have to do an active selection. It's going to do the full change versus.
Yeah, I get so confused. We have another panel that does exactly the same thing. That's better.
Aaron develops panel, and I remember them.
Yes. Right. Yes. So in this case, you can see that we're basically doing a differential expression analysis between these two selections. So the active selection and the saved selection that we saved earlier. And let's have a look. If we colored by, what is this? HLA whatever, HLA DRA, right? Yeah, you can see it. Okay, that makes sense, right? Because this guy is upregulated compared to this guy, and you can do you can do it the other way around as well, and you can look for I guess these are T cells. And then we got this custom heat map as well, and basically in this case, this heat map just shows the the top differentially expressed genes. And I think if I keep on making selections, the heat map will get more and more interesting. So if I save this one, and then I make like another selection here, what will happen is that this heat map will now have basically the log fold change of the active selection, which is this one without the number, against selection number 1 and selection number 2, right? So we have, basically, in this case, this heat map it's not very pretty, but you could just stack this up with more and more selections to figure out, which are your top genes that differentiate one cluster against, any number of other specified clusters. So that's another example of a custom, of a few custom panels that you could play with. And of course, so these are all sort of like, quick and dirty panels that you just want to do something custom. You can just whip up one of these functions really easily, just stick it into, use the create custom table, create custom plot functions to just create an instance of those panels and then just stick them into your app. But later on, actually, if you attend my talk on Friday, I'll talk about how we have a more formal system to set up properly engineered panels. So one of the things about these panels is that they can't actually talk back, right? You can send information to them, but you can't send information out. Like, for example, if I click here, like, there's not even an option to use this selection, right? To color the genes by. That's why I had to manually type HLA DRA rather than just where was it? I guess it's different now because I added this other thing. But I had to type the gene name rather than actually being able to click on it and having this automatically update. So some of the other panels, if you set up a panel sort of properly, and you'll hear what properly is on the Friday talk. But if you set up a panel properly, you can actually get your custom panels to talk back to the default panels, and in fact to any other panel in the system, right? So that your panel can interact with if 10 different people wrote panels that fit into iSEE, they could all interact with each other and send information between each other, and that's a pretty powerful system. Don't know if I had anything else to talk about. Reproducibility, I think someone already mentioned that. I think Fed already covered that a bit, but yeah. So I mean, it's pretty cool, right? You can just you can just reproduce the state exactly as it was before. And usually when I try to do this, I usually don't or sometimes, but I usually don't write it all from scratch, right? Especially if you have brushes, right? So if I want to, you can, if you want, exactly reproduce the brushes as they're shown here. And the information to do that, this thing is in the way. The information to do that is all in the panel settings, right? And this is what I mean by you don't have to, you're not expected to actually regurgitate all of this, right? You just copy and paste it and stick it somewhere, and next time you run the app, you just put the initial, you just run the script and then you put the initial in the iSEE call, and that's really just it, right? Because I mean, I'm not going to write all of this, right? I just copy and paste it.
Aaron, I think we should take a couple of questions before we finish. We're almost at time. So I think the top question is about doing cross-sample feature comparisons and group comparisons, differential expression results. I think we've kind of covered that here. We've shown that you can actually do differential expression analysis in custom panels. And if you put the differential expression results in the raw data, you can visualize them as anything else.
Yeah, definitely. Yeah, so if you've already computed them, then you just stick it in. Yeah, that's right. You just stick it in the raw data. But if you and in fact, I guess we don't have it set up here, but we also have plots for volcano. We also have dedicated panels, as in properly written panels for volcano plots and MA plots, right? That you can show pre-computed results. And as we saw, you can also execute any arbitrary differential expression analysis just by changing how you generate statistics for the custom table.
And if the question is where it's in this ICU package that kind of expands the features, the basic features of iSEE.
Yeah, so that was another question. What was the differences between iSEE and ICU?
Yeah, so iSEE provides the core infrastructure, and ICU at the moment is kind of a tidyverse type package that adds extra panels on top of it. Ultimately, we might split the ICU package into sub packages, each of them with a family of panels to try and keep things tightly related and make it more manageable to update one panel without affecting the others.
Switch from universe to galaxies, probably.
So there's a question on changing the color scales, which you can do, and there is a vignette on that.
Yeah, there is a vignette. So look for something called the experiment color map. Yeah, and you can literally overwrite individual covariates. If you say that, or individual assays, oh, I want my top hat to be my top hat counts to be colored from blue to red, going through white, and I want my log counts to go through the Viridis color scale. You can actually fine-tune every single covariate in your data set.
So what else? I mean, whatever we don't answer here now, we just put maybe in the Slack channel. Mm-hmm. Yeah, so maybe we should mention that. I mean, if you have other questions, there is an iSEE Slack channel that you're free to join, or also the Biosec 2020 workshops channel if there are specific questions about this workshop. Right? So I don't know where is the best place to put this, but maybe in the iSEE channel, or we can put them in the chats and reply to them there.
I just stole the screen from Aaron, so I'll mention a couple just in the last minute or so. So because it's JavaScript based, you can do weird and wonderful things about it. So for everyone who stayed until the end, we have some bonus slides. And we do have voice recognition enabled. So for privacy, we disable by default, and it only works in Google Chrome because it's using the Google Cloud Engine to do your voice recognition. There is a YouTube video that I'm not going to play now for the sake of time, but it's in the online presentation and it's pretty fun. So you can actually cook while you're doing your single-cell analysis. It's robust to egg crack. It really is amazing.
And French accent.
And French accent, yeah, exactly. So, and it's actually supporting the new panels as well. So you can call your panel whatever you want. New panel, it knows how to learn and recognize new panel names. It's just mental. And yeah, and JavaScript, so that was just a warning I had at the start. So you can enable bugs in iSEE. So it is robust to bugs.
This is the only way where you can see bugs in iSEE.
Exactly, and you have to enable them, and you can control how many they are actually. So I put, I think, 10 spiders and 50 flies, which, yeah, it's really highly configurable as an app. How many bugs you want, what kind of bugs you want, and yeah, there we go.
If you want, guys, there are stickers that we are happy to send around. So these are even the hollow version of it. So I don't know, the background is actually messing around, but yes, if there's space on your laptops, let us know.
I'm just muting it, but you got the subtitle. No, it's not the same thing with the subtitles. You really have to hear it, right? But there we go. So
I think that's it.
That's
Great.
it.
Yeah, thanks everyone for joining. Yeah, if you have other questions, then we'll put the answer to the remaining questions as well.